Yersinia pestis and Bacillus anthracus are Category A bacterial pathogens that are the causative agents of the plague and anthrax, respectively. Although the natural occurrence of both diseases is now relatively rare, the possibility of terrorist groups using these pathogens as a bioweapon is real. This video demonstrates a method to detect and identify Yersinia pestis. First, the microorganism is grown on agar plates and in liquid media. Next, a Y. pestis-specific reporter phage is added to the culture. The reporter phage harbors the genes encoding bacterial luciferase, LuxAB, in its genome. If Y. pestis cells are present, the reporter phage will specifically bind to the cell, inject its recombinant DNA carrying the LuxAB genes, and will use the host's transcriptional and translational machinery to transcribe and translate the LuxAB reporter genes. Results are obtained that show the samples have acquired a bioluminescent phenotype, indicating the unknown microorganism has been positively identified as Y. pestis.
The main advantage of this technique over existing methods, like the traditional phage lysis assay, is that the time required to detect and identify Yersinia pestis is minutes instead of days. This method can help to rapidly identify and detect the presence of Yersinia pestis using isolated cultures or directly from complex clinical or environmental samples. The implications of this technique extend towards the diagnosis of the plague because the disease is usually fatal if not treated within the first 24 hours of symptom onset. Thus, the timely identification of Yersinia pestis is essential for a positive prognosis. Since phages exist for nearly all bacterial species, the methodology can potentially be adapted for any species that is medically, environmentally,